The doctor is happy that you found that the treatment produces an absolute risk reduction of 0.03. However, the doctor is worried about how the patients were assigned to the treatment and control groups. For instance, the doctor explains that if the patients who were assigned to the treatment were mostly young and healthy, while those who were assigned to not get treatment were mostly old and sick, then we would see that the absolute risk in the treated group was smaller just because the young healthy patients are less likely to get a heart attack in the first place. The doctor fears that this possible selection bias would bias the results here into showing that the treatment was effective when in reality it was not having any effect. You tell the doctor not to fear because you have randomly assigned the patients to the treatment and control groups. You show the doctor that the patients in both groups look very similar. When we look at the mean and the standard deviation in age and blood pressure values, we see that they're quite similar in both the treated and in the control group. The setup of a medical experiment where we randomly allocate subjects to two or more groups, treat them differently, and then compare them with respect to a measured response is called a randomized controlled trial, or RCT. The doctor is very satisfied by a random assignment of patients into the two groups. However, the doctor now asks you whether it could have been possible that the treatment really had no effect, and the observed effect of the treatment, which is 0.03 ARR, was really by chance. Now, when reporting the treatment effect, it is also common to report its associated p-value to convey the statistical significance of the results. Here's what a p-value of smaller than 0.001 says. It says that there is less than a 0.1% probability that we would observe a difference in risk of 0.03 or more if the treatment actually has zero effect. Because the p-value is small, we thus call this result statistically significant. The p-value is going to be a function of the number of patients we have within each group. If the number of patients in each group was 100, we get a high p-value and our result would not be considered significant. But when the number of patients is 1,000 in each group, we would have a small p-value and the result would be considered significant. Don't worry too much about how these p-values are generated. All that's important to understand here is that we can convey the significance of our results using p-values. The absolute risk reduction of 0.03 has a more interpretable definition in another related measure of treatment effect, which is the number needed to treat, or the NNT. Now, the NNT is simply the reciprocal of the ARR, so it's 1 over 0.03. And the number needed to treat tells us the number of people who need to receive the treatment in order to benefit one of them. In our case, an NNT of 33.3 means that if we treat 33.3 people, we will save one person from a heart attack.